And now finally we are going to see that the action potentials in different parts of the heart, they differ in shape. Okay, and this is because of the fact that different tissues of the heart, different cells of the heart, they have different membrane potentials, resting membrane potentials, and different shapes of the plateau and different shapes of the action potentials. So let's see how they differ in shapes. That we will go through each and every part of the heart and see what is the shape of the action potential in that very part of the heart. So let's start from the SA node. Okay, we have already discussed the action potentials in the SA node, and we have already discussed in the action potentials in the ventricular muscle fibers. But there are other parts in the in the heart, like there are atrial muscles, there is an AV node, there is bundle of His, there are Purkinje fibers. So let's see what is the shape of action potential in the different areas of the heart. So let's start. Number one, SA node. You see, this is the action potential, the SA node. We have already discussed that. Okay, so in the SA node, you remember that there is no plateau phase. Okay, and this spike or this phase 0 is because of the opening of the voltage gated calcium channels. Okay, and this is the repolarization. Okay, and this is the RMP, which is not stable, unstable RMP, or the pacemaker potential. Okay, the next is atrial muscle fibers. Atrial muscle fibers having all the 5 phases. Okay, phase 0, 1, 2, 3 and 4. So there is a plateau present because these are the muscle contractile fibers, atrial muscles. Okay, and then the next one is that of AV node. Again, you see in the AV node the plateau is not very significant, you see, and this is also the here the RMP is not stable. The specialized excitatory and conductive system of the heart has got the property of automaticity and arrhythmicity, so they have an unstable action potential, while the atrial muscles and the ventricular muscles, they are the contractile fibers, so they have a stable resting membrane potential. Now the next is about the bundle of His. As you can see, this is the action potential generated in the bundle of His. Okay, so you can see the shapes of action potentials, they vary from tissue to tissue. Some of them have a short refractive period, some of them have long refractive period. So you see here in the bundle of His, the refractive period is quite long, if you compare that with the rest of the action potential. So sometimes the refractive period is shorter, sometimes it is longer. Okay, sometimes the plateau is there, sometimes the plateau is not there, sometimes the RMP is stable, and sometimes the RMP is not stable. Okay, now you see this is these are the left and right bundle branches, left and here you can see the refractive period is even much longer. Okay, then you see Purkinje fibers. Okay, the Purkinje fibers. Okay, this is the action potential produced in the Purkinje fibers. And finally you can see the ventricular muscle fibers. Okay, the ventricular muscle fibers, as you can see that. Okay, this is another one from the ventricular muscle fibers, as you can see that. So from this you can see that the refractive periods and the shape, the entire shape of the the action potential in different parts of the heart varies from tissue to tissue. Okay, so if we compare the ventricular muscles with the atrial muscle fibers, okay, you can see the duration of the action potential in the ventricle is longer than the duration of the atrium. In other words, you can say that the refractive period of the atrium is shorter than the refractive period of the ventricles, though they have all the 5 phases: 0, 1, 2, 3, 4. In atrial muscles we do have 0, 1, 2, 3, 4. Plateau is present in both. Phase 2 is present here, phase 2 is present here, but the duration is different. Here the duration is a bit shorter, here the duration is a bit longer. But if you compare these two action potentials with the action potential in the SA node, you will see that the phase 1 and 2 is missing here in the SA node. Okay, and the next difference is that the phase 4 over here, the phase 4 is quite stable, but here the phase 4 is unstable, which is known as the pacemaker potential because of the presence of the sodium leak channel. So I hope you appreciate these differences. Okay, and all the cardiac muscle fibers are interconnected through gap junctions. So when one cardiac muscle fiber is excited, the action potential passes from one tissue to another, from one cell to another cell, and the whole cardiac muscle is whole heart is excited in a simultaneously. Okay.